Good evening, viewer. Welcome to our FTV English verse and here's a breaking news for our viewer. Oregon Health and Science University, in collaboration with Oregon State University, has discovered the structural, organization, and protein components of a lipid transfer complex known as LPD thread. Findings show that LPD thread contains an internal tunnel lined with lipid molecules, suggesting a possible mechanism for large-scale lipid movement between cellular membranes. Cells must constantly manage the structure and makeup of their membranes, which rely heavily on lipids produced in the endoplasmic reticulum, and these lipids cannot freely float through the cytoplasm due to their hydrophobic nature. Lipid transport proteins have been shown to supple small numbers of lipid molecules between compartments. A distinct group called bridge-like lipid transport proteins may support bulk lipid transfer by forming long tunnel-like structures that span between organelles, and structural analysis of these proteins has remained limited due to their size and biochemical complexity. In their study, Structural Basis of Lipid Transfer by a Bridge-like Lipid Transfer Proteins, published in Nature, researcher performed cryogenic electron microscope and mass spectrometry on native LPD thread isolated from transgenic canohabitids elegans to reveal the subunit composition and structural details of the complex. Researchers engineered two strands of C. elegans with fluorescent and epitope tags placed at either end of the endogenous lipid thread gene. The C terminally targeted strain, which retained normal developmental and cold stage phenotypes, was used to isolate the native LPD thread complex. Proteins were purified from approximately 60 million worms to generate sufficient material for structural and biochemical analysis. Purification was performed using fluorescence-based site exclusion chromatography. Mass spectrometry identified peptides spanning the full length 4022 amino acid LPD thread sequence. Amazing, sir. Bow down. Behind this report, FTV English, Delhi, Bharat.